Hello po, welcome po sa Makeup Kit.
Transformation and selection of bacteria are key steps in DNA cloning. DNA cloning is the process of making many copies of a specific piece of DNA, such as a gene. The copies are often made in bacteria. In a typical cloning experiment, researchers first insert a piece of DNA, such as a gene, into a circular piece of DNA called a plasmid. This step uses restriction enzymes and DNA ligase and is called a ligation. After a ligation, the next step is to transfer the DNA into bacteria in a process called transformation. Then, we can use antibiotic selection and DNA analysis method to identify bacteria that contain the plasmid we're looking for. Steps of bacterial transformation and selection. Here is a typical procedure for transforming and selecting bacteria. 1. Specially prepared bacteria are mixed with DNA, example from a ligation. 2. The bacteria are given a heat shock, which causes some of them to take up a plasmid. How does that work? The basic answer is that a heat shock makes the bacterial membrane more permeable to DNA molecules, such as plasmids. It appears that the heat shock causes the formation of pores in the bacterial membrane through which the DNA molecules can pass. 3. Plasmids used in cloning contain an antibiotic-resistant gene. Thus, all of the bacteria are placed on an antibiotic plate to select for ones that took up a plasmid. 4. Bacteria without a plasmid die. Each bacterium with a plasmid gives rise to a cluster of identical plasmid-containing bacteria called a colony. 5. Several colonies are checked to identify one with the right plasmid, example by PCR or registration digest. 6. A colony containing the right plasmid is grown in bulk and used for plasmid or protein production. So why do we need to check colonies? The bacteria that make colonies should all contain a plasmid, which provides antibiotic resistance. However, it's not necessarily the case that all of the plasmid-containing colonies will have the same plasmid. How does that work? When we cut and paste DNA, it's often possible for side products to form in addition to the plasmid we intend to build. For instance, when we try to insert a gene into a plasmid using a particular restriction enzyme, we may get some cases where the plasmid closes back up without taking in the game, and other cases where the gene goes in backwards. Why does it matter if a gene goes into a plasmid backwards? In some cases, it doesn't. However, if we want to express the gene in bacteria to make a protein, the gene must point in the right direction relative to the promoter or control sequence that drives gene expression. If the gene were backwards, the wrong strand of DNA would be transcribed and no protein would be made. Because of these possibilities, it's important to collect plasmid DNA from each colony and check to see if it matches the plasmid we were trying to build. Restriction digest, PCR, and DNA sequencing are commonly used to analyze plasmid DNA from bacterial colonies. Protein production in bacteria. Suppose that we identify a colony with a good plasmid. What happens next? What's the point of all the transforming, selecting, and analyzing? Possibility 1. Bacteria is equal to plasmid factories. In some cases, bacteria are simply used as plasmid factories, making lots of plasmid DNA. The plasmid DNA might be used in further DNA cloning steps, example, to build more complex plasmids, or in various types of experiments. In some cases, plasmids are directly used for practical purposes. For instance, plasmids were used to deliver a human gene to lung tissue in a recent gene therapy clinical trial for patients with a genetic disorder cystic fibrosis. Possibility 2. Bacteria is equal to protein factories. In other cases, bacteria may be used as protein factories. If a plasmid contains the right control sequences, bacteria can be induced to express the gene it contains when a chemical sign is added. Expression of the gene leads to production of mRNA, which is translated into protein. The bacteria can then be lysed or split open to release the protein. Bacteria contain many proteins and macromolecules. Because of this, the newly made protein needs to be purified, separated from the other proteins and macromolecules before it can be used.